All right, so now that we've gotten through the general senses, we're going to get into some of our special senses. And for this video, we're going to focus on two. We're going to look at olfaction and gustation. So we're going to talk about the sense of smell and the basic anatomy for olfactory sensory neurons and whatnot. We're going to talk about olfactory transduction and the neural pathway that's involved with that, as well as getting into our sense of taste and the basic anatomy of taste buds, get into gustatory transduction and the neural pathway involved in gustation. So let's start with olfaction. So with olfaction, we're talking about smell, and smell is going to be using chemoreceptors. So our whole idea here is that we are going to detect molecules that have actually entered into our airways, right? These molecules have gotten stuck in the mucus inside of our nose, right, inside of those airways, and they actually join up with proteins to be detected by these chemoreceptors. Your sense of smell is extremely good. Your nose contains about 100 million receptors when we have the ability to detect about 380 different odorant receptors for that. So 100 million total receptors in about 380 different categories. So our sense of smell is very fine-tuned in comparison to some other ones. The crazy thing though is that compared to other animals, our sense of smell is actually very weak, where you look at something like a dog and these numbers jump significantly, several times over what the human has. These sensory neurons, these olfactory sensory neurons, are going to project from the olfactory bulb. We see the olfactory bulb here kind of projecting out towards the ridge of the nose, basically internally right inside the skull, and we see that there are little holes in what's called the cribriform plate of the ethmoid bone. These neurons are actually going to send themselves down through those holes into the nasal cavity. So let's talk about some of these cells. So we have a bunch of different cell types hanging out in here. We have the olfactory sensory neurons, which we've already mentioned. They're the ones who are detecting those chemicals that have been bound to proteins in the mucus via their dendrites. The mitral cells or mitral cells, these are going to be cells that form the olfactory tract. They're going to be the neurons that carry the impulses to the brain, so they connect the olfactory sensory neurons to the central nervous system. And they have connections that extend from the olfactory tract up to the temporal lobe, the insula, the limbic system, and the hypothalamus. And we'll talk about that in a little bit. Supporting cells are going to provide nourishment and support for these sensory neurons, and then mucus glands produce the mucus that's needed so that the odor molecules can actually stick and dissolve in the mucus to bind with the proteins for detection. So olfactory sensory neurons, these are the neurons that are going to synapse with those mitral cells, which are also neurons found in that olfactory bulb. The olfactory sensory neurons project down. So if we're looking over here, right, here's our olfactory tract, the mitral cells coming down kind of at that 90-degree angle here. We see the synapse, right, with the olfactory sensory neurons that extend down through the ethmoid bone and down all the way until they have their little modified dendrites, right, sticking out in between the epithelial tissue we find in the nose, in that nasal cavity, right? So we have little olfactory cilia that are going to be, we have mucus cells that are going to be producing mucus, and all of those little dendrites are hanging out in the mucus. So the chemoreceptors that are on the dendrites, they sense those different chemicals, right? And again, the chemoreceptors, they sense different kind of odorants. They detect different kinds of molecules. They can only detect certain ones. It's not like an all or nothing, like you detect any of those like 380 we talked about. They're going to specialize, but we have so many of them, right? And it's the combination of those getting activated that gives us a specific scent, right? Those mitral cells that connect that synapse with the olfactory sensory neurons, right, they're going to form the olfactory tract, which is going to be our first cranial nerve, the olfactory nerve, which then goes towards the insula, the temporal lobe, as well as the limbic system and hypothalamus, which is because it has that direct connection to things like the limbic system and whatnot. This is one reason why your sense of smell is so strongly tied to memory, and why you're smelling something can trigger memories stronger than other senses can. Now, gustation, this is your sense of taste. So gustation is just the fancy term for that. Again, we are looking at chemoreceptors, and there are 5 primary tastes. So we see a difference here in terms of strength or in terms of diversity of receptors in olfaction versus gustation. In fact, your sense of taste, because there's only 5 primary tastes, is generally pretty bland, and that the fine detail, the nuance you get when you taste food and whatnot actually comes from your sense of smell. It comes from olfaction. So gustation and olfaction are actually closely tied to each other when it comes to our overall perception of something, because gustation gives you the basics, gives you the baseline of how something tastes, but your olfaction, your sense of smell, is going to give you all the nuance that lets you tell a lemon from an orange apart. But we have 5 basic tastes: sour, sweet, bitter, salty, and umami, sometimes called savory. There are some things up in the air in determining whether or not we can taste fats, if that should be considered a 6th sense of taste, but we haven't quite gotten there to solidify it yet. You may have also heard of the tongue maps and whatnot, that certain parts of your tongue only taste this sour or only taste bitter, and that has been debunked recently. So that's not something that you should commit to memory of any sort. We do have different types of papilla that we can find taste buds on, and there may be different concentrations of these taste buds in different areas. However, that map that you saw when you were in elementary school does not necessarily hold true. So let's talk about taste and sensing the different types of molecules that are dissolved in your saliva when you are eating. We're using gustatory receptors found on the microvilli on what we call taste cells that we find on taste buds, right? So here are the different type of papilla we have, the different shapes. So circumvallate, right? These ones that are kind of rounded and then dive in. Fungiform, right? Kind of round over. Filiform, right? Kind of form little peaks. And then foliate, right? Are more flattened. And we do find these different papilla in different parts of the tongue, right? We find the taste buds kind of extending out from the side there, right? These little kind of extensions you see here. And it's on those taste buds that we find the taste cells, and those taste cells have the receptors that detect these special chemicals, right, that are found in the food we eat. So these receptor cells, it's important to note, these receptor cells are not neurons. This is going to be one of those specialized ones where we have an epithelial cell that does the detecting and then communicates with a neuron nearby. Right, so we have the little microvilli, they extend through the pore, they detect the chemicals in the food, and when they contact that chemical, right, that's when they're going to release a neurotransmitter to trigger a sensory neuron nearby. But the sensory neuron in this case, with gustation, does not specifically detect, it doesn't directly detect the chemical. The gustatory receptor cells do, and then these cells communicate that with the sensory neuron. So if we look here, we kind of see that, right? We see the little taste hairs, right? We see little microvilli that extend out of that pore, right? We see the cells themselves, the gustatory cells, right, down around there. We see transitional cells in between and some basal cells, but then we see that we connect, right, to the sensory neuron down at the base. But it's a different cell that is extending up and actually tasting the things, right? Detecting those chemicals in the foods we eat. So let's talk about gustatory transduction. Each taste cell has taste receptors specific to a certain type of taste. It's not an all or nothing. They can either detect salty, sour, sweet, umami, or bitter, but they can't detect multiples of those necessarily. These taste receptors are found on those microvilli, again on the apical surface of the top side of our taste cell. So again, the taste cells are the ones here in this kind of pink beige color with the little microvilli, or what we call taste hairs, extending out, and they're the ones who are going to make that detection. Then when they make that detection, there's a couple ways it can happen depending on what we can detect. If we are a taste receptor cell that detects something that is salty, we will undergo depolarization when we come into contact with sodium ions. right? And that makes sense because sodium ions are part of table salt. And so when something is really salty, right, you're tasting part of that salt, right? It's going to cause a depolarization inside of the taste cell though. With sour, we are going to be depolarized by hydrogen ions, right? So same kind of process, except different ion causes depolarization. And we see a lot of things that are sour are acidic, and that hydrogen ion will be released by something that is acidic. With sweet, umami, and bitter, we have a different method. We're not being depolarized by an ion anymore. Instead, we see G proteins being activated when we come into contact with that trigger, right, with that food source. If we're sweet, our G proteins are activated by monosaccharides or sugars. If it is umami, the G proteins are activated by glutamate, so by amino acids, which is why protein heavy, so like meats and whatnot, tend to be very savory. Bitter is one that's a little bit different. It is activated, the G proteins are activated, but there's a wide variety of things that can activate these. I'm only going to focus on one, and that's alkaloids. So things that are alkaloids tend to trigger the G proteins that are in the bitter ones. Right, but there are a couple different things. I'm not going to have you worry about the other stuff, just remember alkaloids. So what's the gustatory pathway? These impulses from our taste cells are carried by 3 different cranial nerves, depending on where on the tongue we're looking. I'm not going to make you memorize that part, but you will have to know which nerves are involved with the gustatory pathway. And of the 3, we have the facial nerve, the glossopharyngeal nerve, and the vagus nerve. Right, so these are the 3 that are involved with taking that information, right? They're going to have those sensory neurons that are going to take that information from the taste receptor cells, right, and carry it forward to the central nervous system. So we still have 3 neurons in order to get to the brain from the mouth, basically. The first order neuron is going to be what is going to synapse with the taste cells and be carried along through the medulla oblongata. The second order neurons will carry us from the medulla oblongata to the thalamus, and then the third order neurons will go from the thalamus to the insula, right, where we can again be tied to taste and that kind of inner portion, that inner lobe called the insula.